Thank you all for coming to listen to this today, and thank you to the organizers for giving me the opportunity to present today. I'm really excited to talk about this project I've worked on for a couple of years now. You will see in a moment that the biology is very grounded in developmental biology and stem cell biology, but when we get to the modeling, you'll see there's also a lot of population genetics at play. So I hope that there's a little bit for everyone with this project. So I'm going to start by giving a very broad biological overview of the problem that I work on. So as I'm sure you all know, our bodies consist of lots and lots of different cells, and in order to stay alive, it's necessary for our cells to be constantly dividing and constantly dying to stay alive. And in the process of cell division, there is the capacity for cells to acquire mutations through division. And I'm going to represent that in this picture by changing one of my cells to be blue. And so the color here is simply to say that in the process of dividing, one of these cells had a daughter cell that had picked up a mutation. And so it's a little bit different from the rest of the cells in the tissue. And most mutations don't have a significant role in the cell's behavior, but they're nonetheless there, and they can persist in our tissue. Sometimes, by random chance or by selection, these mutations have the ability to expand in the tissue simply by being lucky and getting to divide more and having more daughter cells. And so I'm going to represent that here by showing that the blue mutation has been able to spread. And when this happens, we call this a clone. So a clone is simply a group of cells that all share a common ancestor, and in this case, they all also share a common mutation. And the picture I want to paint for you here is that our tissues don't consist of these sparse mutations with some clones appearing here and there, but really our tissues are full of these different heterogeneous types of cells that all come from different mutations and that all have the ability to expand. But one thing that is observed in lots and lots of different organisms, as well as lots of different types of tissue, is that as we age, as organisms age, some clones begin to dominate others, and in general, we see fewer of these clones. And so maybe the picture changes, and some of the clones have grown, and some of them have been wiped out. And so this is a process that I'm very interested in understanding, is what is the process that takes us from a diverse group of different clones to a more coarse mosaic, I guess, of the cells. And as I said before, a lot of these mutations are neutral and don't affect the tissue, but there is also a lot of understanding of how this clonality is related to the onset of age-related conditions, and in particular, cancer. So to understand this process, it's very important to look at stem cells, because these are the cells that are responsible for both developing and maintaining specialized tissue in our bodies. And so there are lots of different types of stem cells. For example, there are hematopoietic stem cells, and these are the cells responsible for producing not only our cardiovascular system, like red blood cells, but also our immune system. And another example are spermatogonial stem cells. These are the stem cells that reside in the testes of male organisms and are responsible for producing a constant supply of sperm throughout adulthood. And it's these cells on the right, the spermatogonial stem cells, that I'm going to be focusing on for this project, as you'll see in a moment. One difficulty in studying stem cell populations is that they tend to be in very inaccessible locations in our tissues, and also there tend to be very few of them. And so what that means is that if we wanted to look at them directly, that usually involves either damaging a tissue or killing an organism. What my group does is we work with the zebrafish, and in particular the zebrafish testis, as a model system to study stem cell dynamics. And the reason for that is, while it is still difficult to access the stem cells in the testes of these fish, it is very easy to take sperm from these fish to get an indirect view of what the stem cells are doing across the fish's adult life. And so my role in this project is to use mathematical modeling of population genetics in combination with experimental data I'll describe in a moment, to understand how these clonal dynamics are unfolding in this model system. So before we get to the model, let me talk about the experimental setup that we use. So early on in a fish's life, specifically right after the embryo has been fertilized, so I'm talking about 0 to 4 hours post-fertilization here, there's a small group of cells that are sitting in the embryo, and these are the cells destined to become the testis in the adult fish, assuming this fish becomes a male. And so these are called primordial germ cells. What we do at this early embryonic stage is use CRISPR gene editing to insert a unique genetic label into each of these primordial germ cells. So this is an edit that is not going to change the cell's behavior in any way, but it is a marker that we'll be able to trace back later using sequencing. And so in this picture here, I'm going to represent that by changing each of my cells to be a different color. as a way of saying that we can now distinguish these cells when we do sequencing later. And so once we've done this labeling, the fish is going to continue to develop, and by the time it reaches adulthood, which is about 3 months later, each of those labeled cells has become a clone of cells in the adult testis of this fish, and that barcode has importantly been inherited throughout that entire process. And what we can begin to do at this point is we can now sample sperm from the fish periodically and get a sense of how big are each of these clones in the testis, and then we can do this in a way that isn't hurting the fish, not too much. And so here's a picture of what that data looks like. So what I want you to see is that the x-axis is representing time. So we sample sperm from the fish about once a month. And then what the y-axis is telling you is each color is telling you the proportion of the sperm that we collected that comes from a given clone. And so what you see is at 5 months old, about 80% of the sperm we collected belongs to this hot pink clone. And as the fish gets older, we see that that clone is being represented less and less and less as the fish ages. And at the same time, you see this orange clone that started off small and is now being seen to expand. And along the way, there are lots of other clones that are fluctuating up and down in their size. And we've done this not just with a single fish, but with a cohort of about 30 different fish. And so each fish tells you a slightly different story, and you can see slightly different dynamics from fish to fish. I'll make the note that the color scheme is the same between all of the fish. I start with hot pink and go down from there, but we're imagining that each of these labels are different from one another. And so you shouldn't necessarily correspond what the hot pink clone is doing in one fish with what's going on in another fish. And so there are a few observations I want to make and emphasize to you all right now. The first thing I want to note is that only a few clones are dominating the sperm pool of these fish. When we do our CRISPR labeling, we're anticipating that this is going to label about 10 to 50 primordial germ cells. But what we see in these adult fish is that maybe 5 of those clones are dominating most of the sperm pool, and then there are maybe 10 to 12 total clones. The second thing that I find very interesting is that small clones are persisting. This isn't in line with the picture I had shown you earlier with clones getting wiped out by clonal expansion. And I think that's very interesting as well. We see the small clones changing very, very little in size, while the big clones, they're fluctuating a bit more, it seems, relatively speaking. And then the last thing I want to point out is we do see consistent changes in the size of clones over time, like what I said with the example at the top left. We see that pink clone consistently shrinking while that orange clone is consistently growing. And so how can I take these observations and start thinking about how I can maybe test hypotheses about what is driving the clonal dynamics we see? So the first thing I want to understand is how strong is genetic drift in the clonal dynamics we see here. So we see that the pink clone has the capacity to change a great deal, and maybe we believe that the small clones are undergoing less drift. How can I quantify that rate of drift and say for sure whether there is evidence that clones are behaving differently from one another? And then the second thing I want to ask is there evidence of selection? And so what I mean by that is I want to ask, with my pink clone I had at the top left, is that clone shrinking because it got unlucky due simply to some kind of neutral drift? Or is it shrinking because it actually had some kind of disadvantage compared to the other clones? Or more recently, maybe it is possible that the orange clone has some advantage that is allowing it to expand more relative to the pink clone. And so these are the kinds of questions that I hope to be able to address quantitatively using a model. So before I get into that, what is the precedent? What do we already know about clonal drift in other organisms? And the answer is it varies a bit, and it depends a little bit on the biology of how the testis and organs work in other systems. So what I can tell you is that for the testes of Drosophila, the testes of these organisms tend to have a bit less drift than we see in other organisms. And the reason for that is there's a particular structure to the testis that allows, that emphasizes what are called asymmetric stem cell divisions in the testis. So these are stem cell divisions in which one daughter cell produces a stem cell, and one of the daughter cells goes on a fate to produce a bunch of sperm. And under this kind of division, it becomes very hard for a clone to expand, because if our testis is doing only these kinds of divisions, there's not much of an opportunity for a clone to expand in size over time. We started with one stem cell that's dividing, and we ended with one stem cell that's remaining in our tissue. On the other hand, if you were to look at a mouse, they have a slightly different structure to their testis. And so there's, instead of these asymmetric cell divisions, a balance between what are called symmetric cell divisions that are producing 2 stem cells and symmetric divisions that are producing a lot of sperm. And so what we can see in mouse testes is a lot of drift because there is an opportunity stochastically for a clone to increase in size through these symmetric divisions, and at the same time, there's also an opportunity for stem cell populations to shrink due to these divisions. And now, how about selection? Is there a precedence for selection happening in the clones of organisms? And the answer is yes. This is seen primarily in humans through what are called paternal age effect mutations, which I'll talk about on the next slide. So there's this very important pathway in cell proliferation and growth and survival called the RTK-RAS-MAPK pathway. And all I really want you to know from this picture is that there's a group of genes that are very important in this red box here, and the ones highlighted in red specifically are known to be important mutations as it relates to clonal expansion in humans. So if a human has a mutation in any of these highlighted genes that's associated with some kind of fitness advantage in the stem cells of their testis, and that leads to a clonal expansion as they age. And these are pretty well-studied mutations because they're also very strongly correlated with birth defects in the offspring of older men. These birth defects come as a consequence of a mutation happening, that clone expanding in their testes, and then all of the sperm they're producing now also carry these mutations. And so we do have some evidence of selection happening in these systems as well. And so with that said, what am I contributing by working with the zebrafish here? I think it's really about this data here. It's very hard with mice and humans to get a time series data like this. And so what I'm hoping to do with this project is really take this type of data that I think is really, really exciting and really interesting to better understand how clonal dynamics are actually unfolding. So the way I'm going to set this up with a model is what's called a hidden Markov model. So there are a few steps that go into this model. I first need to have some representation of what does a testis look like in my zebrafish, and then I'm going to have some stochastic rule for how the stem cell populations are going to change in time inside this testis. And I'm going to call these my hidden testis dynamics. And the reason I'm calling them hidden is because I never have access to data about the testis state directly. Instead, what I have are indirect measurements of the testis state through my sperm sampling, and so that's the data I have available to me. I'm interested in understanding a process in the top row here with my testis, but my data is some kind of indirect measurement from my sperm. And so what I need to tell you is I need to tell you what is the rule I'm using to describe the testis dynamics and how am I going to represent my sperm sample as some observation of the testis state. For the process at the top, I'm going to use what's called a Moran process to describe clonal dynamics. This is a model that originated in population genetics to describe changes in allele frequencies in organisms within a species over time. And I'm using it here to describe how how clonal identity and clonal frequencies are changing over time in the lifespan of my fish. And so I have this little picture to kind of illustrate what this looks like. What a Moran process says is that I have a group of cells, so here I just have them nicely laid out in a grid, and then each of these cells is on an exponential timer to undergo a differentiation event. And so maybe what I'm going to say is that this blue cell is stochastically the first one that has hit its exponential timer, and it's going to differentiate, and I'm going to think of that as being removed from the system. And whenever this happens, I want to maintain a constant stem cell population. So another cell randomly is going to undergo a symmetric division and take the spot of the cell that I just differentiated. So this process is a way for these stem cell populations of different clones to stochastically fluctuate up and down and experience genetic drift. I'll let this play out. Cool. So that's the process I'm going to use for the hidden testis dynamics of my stem cells, and at the moment I'm going to assume neutrality, so none of the cells have some kind of inherent advantage or disadvantage over one another. Great. Now, how am I going to go from this process that I'm using to represent genetic drift in the stem cells to observe sperm data? Right now I'm going to do what is the simplest possible solution, which is to take a binomial or multinomial sampling of the testis state and make that my sperm sample. So what I mean by that simply is, let's say that my testis consists 50% of red cells and 50% of blue cells, then I'm going to assume that each sperm has a 50% chance of being red and a 50% chance of being blue. This is probably one of my more controversial modeling decisions because it is a bit removed from the underlying biology. More realistically, I'd like to be able to, when I'm doing my differentiations in my testis level, to keep track of the sperm that's produced and then let that be my observation. The reason that I'm working with binomial sampling for now is it is the simplest possible choice, and it's going to allow me to do very nice likelihood in a moment. And so it's the choice I'm going to stick with for this presentation. With that said, I am working with my group to do some experiments to really make sense of whether this is a good decision. And if it proves to not be a good decision, we can go back and look over that assumption. Okay, so now that I have this model set up, I want to go back to the data and then go back to my question here of are clones undergoing drift, and if so, what is the strength of that drift? And I'm going to, for this presentation, set this up as a kind of hypothesis test. So I'm going to make my null hypothesis that there is no drift at all, and I want to ask, is there significant evidence to the contrary? Is it far more likely that the data you see here is coming about from drift? And I think from this picture, at least with maybe the pink or the orange clone, it seems very clear, yes, there should be drift, and hopefully my model can affirm that. But with these smaller clones, it's not entirely clear if they are behaving according to the same rules. And so I want to start with this question of, is there non-zero drift? What is the likelihood that there is non-zero drift versus zero drift? So let me set this up with a simpler example. So here's a simulation of my model where I just have two clones, and I can tell you some parameters for how I went about simulating this. And what I just want you to know is that when I'm simulating my model, you see some variation from time point to time point. And this is coming about through a combination of possibly genetic drift, if I simulated this with genetic drift or with replacement, but also because of my sampling step that adds a little bit more noise to the data. And so when you look at this, you might see what looks like possibly some kind of trend. So maybe there's drift happening, but it's not entirely clear. Maybe this is just entirely due to noise. And so what I'm going to do is try to predict what rate of drift will be most likely to reproduce this data. And so I'm going to use ML to estimate my drift parameter, where of course by ML, I'm referring to maximum likelihood. So what I'm going to do is I'm going to define this likelihood function, and that's simply the likelihood of seeing this observed data given some specified rate that drift is happening. So this likelihood function evaluated 0 is the probability that if I simulated my model with a drift rate of 0, what's the probability I get this exact data? And the beauty of the way I've set things up with the random sampling with replacement and everything is I can compute this likelihood exactly, which is a very nice thing for me to be doing, especially as a starting point. And so my null hypothesis is going to be that there's no drift at all. And to reject or accept that hypothesis, I'm going to use what's called a likelihood ratio test statistic. So this is really a comparison between the value for the rate of drift that is most likely to produce this data to the likelihood of getting this data with no drift. And if that difference is significantly large, then I will say that there is significant evidence that there's a non-zero rate of drift, and I can kind of reject my null hypothesis. And if this test statistic is too small, then the probability of getting this data with no drift is about what it would be if I did allow for drift, and therefore I shouldn't reject my null. And for this presentation, I'm going to make my cutoff 6 as an arbitrary choice. And this corresponds roughly to a 20-fold difference in the likelihood calculation. The way that I calculate likelihoods, for anyone who's not familiar with hidden Markov models, I use this thing called the forward algorithm. You can imagine if I wanted to calculate some probability of getting this observed data here, the naive approach would be to consider every single possibility of what happens in the testis states and marginalize over that. And that becomes very computationally tricky. But this forward algorithm that's well known in hidden Markov model land is a very computationally efficient way to compute likelihoods in a way that exploits the Markov structure and saves us some time. Yeah. So with this picture here, this is a plot of what that likelihood surface looks like. So the x-axis is my differentiation rate, that parameter r, that says how quickly are cells differentiating and getting replaced. And the y-axis is my log likelihood. And so when I'm doing maximum likelihood, what is the drift rate that is most likely to produce this data? What I can see is that a drift rate of 1.25 is most likely to produce this data, meaning each cell is on average differentiating once every 1.25, or excuse me, wrong way around. There are 1.25 differentiations per time unit on average. And I want to compare that to what's the probability of getting this data with no drift. And I can do that by taking the difference in those likelihoods from my maximum to what happens when the drift rate is 0. That's a difference of about 4.28, so the test statistic is double that. And in this case, my test statistic is above 6, my test statistic is above 6, and I would reject my null and say there is evidence of drift in this simulation. And to give you the correct answer, I simulated this with a drift rate of 1, so we slightly overestimated, but made the correct conclusion in the end. Okay, so what happens if I try to apply this approach to my experimental data? What I'm going to do is I'm going to separate each clone out individually and try to estimate a drift rate. So for example, with my pink clone, I'm going to think about pink clone compared to everyone else and estimate a drift rate of just the pink clone, and do the same for every other clone. There are 2 reasons that I'm doing this. The first is that I think it is very, very interesting to see whether we get similar estimates for all of the different clones, or if there is a lot of variance in the drift rates that are predicted. And then second is for computational reasons. There's a lot of difficulty in computing likelihoods with a ton of clones at once, as that causes an exponential increase in the number of states I have to consider. And so what I estimate, this plot I have here, the bolded value is the rate of drift that is most likely to reproduce this data, and then in parentheses I have my test statistic. And for this fish here, I think the takeaway is kind of what we expected, in that my model is saying with pretty much certainty that, with significance, that all of these clones are experiencing some amount of drift, the exception being there's this very tiny clone down here that's not changing enough to establish significance. And I also want to affirm this thing I had stated a moment ago, where I think that large clones are experiencing more drift. We do, in fact, see that in this picture. And in fact, the same really is the case with a lot of these other fish. The general takeaway is large clones are experiencing more drift, and this is surprising because this shouldn't happen under neutrality. A stem cell should not have awareness of how big its clone is, and therefore I find it surprising that these clones are somehow experiencing more drift as being picked up both by I and also by my model. And another thing that I find interesting that my model shows is that clones that begin large in size tend to decrease in size. So you see with 3 of these fish, the pink clone as a whole is generally shrinking, and this seems to be the case in a lot of our fish. And so maybe there's more to the picture than what I'm saying here. And to get at that, we can again ask a slightly different question of, is there evidence of selection rather than simply neutral drift? And so the way I'm going to represent that in my model, remember earlier, I had this blue cell that had differentiated and a red cell that replaced it. And the way I represent selection here is I'm going to weight the probability that a target clone is going to divide by 1 plus s, where s is a parameter I can specify. That's the selection coefficient. And so I have this new parameter that if it's positive, if s is positive, then I have positive selection for my clone. It is prone to expand. And if s is negative, I'm inclined to shrink. And I can set up a similar kind of hypothesis test with this clone. So my null hypothesis is there's neutrality. This clone doesn't have an advantage. And I can tell you that the likelihood for this particular clone gets maximized at s equals negative 0.19. That is very strong negative selection, which makes sense. We see this clone is shrinking. And we have a test statistic lambda of about 8. And so this might suggest that this clone is in fact somehow under some negative selection, or conversely, the remaining clones are just somehow at some selective advantage compared to our pink clone. This step is a little bit preliminary, and I have some things I want to sort out with the selection question, but this is something that my model is set up to do. And one thing we're hoping to do experimentally, I told you about this pathway earlier, and what we want to do as a next step is be able to introduce some of these mutations into the primordial germ cells of our fish. And we hope that by doing this, we can see selection in action in a case where we expect it to actually occur. So with all that said, I've talked to you about stem cell clonality and why I think it's an interesting concept, and hopefully you all find it interesting as well. And I've talked about this experimental approach we've used to try to study clonal dynamics more directly. And then finally, I've talked about some very simple modeling I've been doing to try to make sense of genetic drift in our data and start to do likelihood and inference on that. I have the great privilege of having both a wonderful math advisor in Fred Adler at the University of Utah, as well as a fantastic biology mentor in Jamie Gagnon. Jenna and Andy are graduate students who are also they contribute a lot to this project as well, and I'm very thankful to them. But if any of this was interesting to you all, I would be delighted to talk to you more. And even if this project is nothing related to what you do, but you are somehow in need of a zebrafish collaborator, I'd be happy to chat as well. So thank you all for listening.
Okay. Okay, let's have a few questions.
Hi, thanks for the great talk. I have a question. So I was wondering, I know you don't want to hurt the zebrafish, but if you ever sort of sacrifice them at the end to get a sense of, how the clonality of the actual stem cells compares to what you see in the sperm readout, because that probably would be a nice way of sort of calibrating your model and answering the question about, differentiation, right? If differentiation is proportional to clone sizes.
Yes, that is exactly one thing that we're doing right now, and we already have those dissected testes. We just need to kind of analyze that data. That's our main approach to making sense of, is our sperm sampling a good representation of the testis, or is there some kind of bias in play? Yes.
Okay, well, a question from the back.
Maybe that's kind of similar to the question that was just asked, and also great talk, very clear. But is there any concern that for some reason some clones would be overrepresented in your sample?
Yes, so when I've talked about selection in this talk, I was referring to selection as a cell being more prone to divide than another, and that allows the clone to expand within the testis. There's also, selection can mean multiple things here when we're thinking about germ cells, and maybe that can be not that the clone is more inclined to divide symmetrically and take over the testis, but that it's more inclined to divide in a way that, or differentiate in a way that just produces more sperm. And this is actually one thing that I suspect could be happening. I mentioned a moment ago with a lot of these big clones that are shrinking very quickly, the hot pink clones. What I think could be happening, if my clicker would work, what I think could be happening here is not that these clones are under some selective disadvantage that's making them shrink in the testis. I think what could be happening is that they're just being called on very early on to differentiate a lot, and that causes them to be overrepresented early on, and then these clones might be burning themselves out. And so what could be happening is the pink clone might be overrepresented early and then underrepresented later. So yeah, this is something I really need to understand better.
Cool, thank you.